the pronunciation was not good. I'm from the Universidad del Desarrollo here in Santiago, Chile. So first, thank you really for the kind invitation. And I'm going to talk to you about something completely different from what we've been listening in the previous talks. And it's about how to model phenotypic variability using actually the different genetic backgrounds of animal models, and how to use these to really translate it into patients. So for different reasons, I've been focused on lysosomal storage disorders, and this is the problem. So for example, these are patients of Gaucher disease, which is caused by loss of function mutations in an enzyme that breaks a lipid within the lysosome. And the problem is this one. Patients can present with a very different symptoms, even though if they are sharing identical mutations. And this is what I'm showing here. I don't want to go into the details of the patients, but as you can see, for example, patient like the patient in the picture D, it presents a very aggressive neurological disease, which comes with very short lifespans. And on the other hand, the patient that it's in A, which receives her medication, she has a normal life and she is a college student. So clearly, the genotype-phenotype correlations are really, really poor. So the title of this paper is Divergent Phenotypes in Gaucher Implicates the Role of Modifiers. So the challenge is how to identify these modifiers. And usually there are different strategies. One strategy is to look for candidate genes. Let's say some patients, they present bone abnormalities, so they go and look if in these patients they have, let's say, variants in genes related to bone homeostasis. Using this kind of approach, at least for Gaucher disease and for other lysosomal storage disorders, we have zero results. Another option it will be to use linkage analysis, and for that you need really a very large families where you have patients presenting with the disease and different clinical symptoms, which are really hard to find. So it has been really difficult to identify modifier genes using this strategy. Another approach, it will be to use genome-wide association studies, or GWAS. And here you need to have a group of patients with one symptom, another with a different one, then to genotype them and compare their genomes. And basically, it's very costly. And for rare disorders, there are not enough patients in the world to assure statistical power. So basically, none of these approaches can be really followed. Instead, I present this strategy in which basically we get animals, in this case mice, from different genetic backgrounds, and we model the disease on them. It can be done by genetic or chemical means. Then we group the strains according to their different phenotypes or responses. The genomes of these guys are already sequenced, so we can just perform the association study. And then the final goal is, of course, to identify genetic signatures that may explain the phenotypes in mice. And this can be used, again, to search for biomarkers in patients. And of course, to design novel therapies for each symptom or sign, okay? And at the end of the day, we would like to predict the phenotypes in mice and humans. So this strategy has several advantages. I already mentioned some of them. The first one, actually, it allows us to develop new animal models for studying the biology of the disease. We have access to the tissues of the animals, so we can really learn about the biology behind, biomics, immunostainings, etc. it is possible to replicate the measurements in genetically identical individuals, so minimizing environmental effects. So within every strain, the genetic variability should be 0, and that's why some people really say that using this kind of strategies, we're minimizing environmental effects. Inbred strains are homozygous for each loci, so it facilitates the bioinformatics. Well, their genomes currently are not only the SNPs, their genomes are fully sequenced and free available. You don't have to spend money on that. And this is a strategy to identify putative modifier or candidate genes that then can be really can be validated in patients using a smaller number of them because you're focusing on candidates. This is due to the false discovery rate corrections, which are smaller. So let me tell you, I mean, the strategy works. These are results from my postdoc, where I was chemically inducing Gaucher disease in 15 different inbred mouse strains. And I started to phenotype the different strains. And here what happened was really remarkable. A group of strains, they presented with very short lifespans, these ones here. And they, as humans, they presented with neurological signs. While other animals, they presented longer lifespans and their brains were healthy. Again, mimicking what happened in humans. So then we decided to carry out the GWAS studies. And for that, we use I would say a very black and white phenotype, which was lifespan. And like that, we identified several loci and putative modifier genes. This is very interesting because we identify, most of them are ion channels or transporters that were not previously related to the disease pathogenesis. And from the lifespan data, most of our candidate genes are genes that are expressed in the brain. So it's telling us really something. We managed also to identify gene signatures that allowed us to predict the lifespan of the animals upon induction of the disease with the drug. And actually currently what we're doing in collaboration with Tim Cox in Cambridge and John Hardy and UCL, we're sequencing Gaucher patients with different disease severity and trying to validate our putative modifier genes. Furthermore, among the top genes, we identified the subunit B of the NMDA receptor that really mediates brain, glutamate cytotoxicity in several brain disorders. And we measure the levels of this transporter in the brain of the short-lived and long-lived animals. And in the short-lived animals, their levels were much higher. We were lucky enough to count with an FDA-approved antagonist of the receptor. So we treated the short-lived animals with the drug. And here you can see the average lifespan of the animals. In red, the untreated, and in green with the drug. So we managed to duplicate, to triplicate the lifespan of the animals depending on the strain. And furthermore, we had access to few biopsies of patients with neurological disease and non-neurological disease. And the neurological patients, they presented also with higher levels of this receptor, really suggesting that we managed to identify a potential therapy by drug repurposing. So this is pretty much the strategy, and I really enjoy it because we're starting from an unbiased phenotype. We managed to identify putative genetic biomarkers, which allowed us to predict the disease severity and also a therapy. So a couple of years ago, 2.5 years ago, I started my lab, and we decided to apply this strategy for other problems, not only in terms of of disease, but also under physiological conditions. I'm interested in identifying putative novel treatment for lysosomal diseases. So here, what we're doing currently, I mean, in most of these lysosomal diseases, patients, they don't present zero activity, they have little activity. And if we manage to increase it by modulating a second gene, that will be a novel therapy. So we're analyzing, for example, the hepatic levels of lysosomal exosaminidase B activity, and naturally the different strains, they present with different activities. And again, we're performing the association studies. We have identified some putative modifier genes for this. Currently, we are studying the mechanism by which this novel gene, it's modifying the activity by overexpressing. We have identified positive correlations. When we overexpress the gene, we get more XB activity. But we're doing this not only for one enzyme, we're doing this for 15 different enzymes. We've been measuring also their substrates. We have the transcriptomes of these strains that are also available. So basically, we're putting all the pieces together and we're building Bayesian networks, which will allow us to identify nodes, which will be the best drug targets to increase the activity of these enzymes. We're very excited about this, very preliminary data about the networks, but it seems it works. So now I'm gonna change gears. I'm gonna tell you a bit another lysosomal disease. This one is Niemann-Pick type C, or NPC. This disease is caused by loss of function mutations in two proteins that are involved in the exit of cholesterol from the lysosome. And it triggers brain liver degeneration. And again, there is a variation in the time of onset and the rate of the progression of the disease. Usually the disease is divided into three different subtypes. The infantile patients survive till they're 2 or 3 years old. The juvenile, the most common one, they live till they're teens. And currently we are identifying an adult onset where patients live for several decades and they show dementia. So we've been lucky enough to have a mouse model of the disease, and this model really mimics the juvenile form. The animals live like for 11 weeks and they present with Purkinje cell death, hippocampus problems, etc. So we were interested in addressing the role of genetic background, and we back cross and we transferred the genetic mutation from the BALB-C background, which it's shown here in this part of the graph. And when we transferred the disease to the C57 background, we observed a reduction, a dramatic reduction in the lifespan of the animals. And when we analyzed their tissues, we identified that these animals presented liver and spleen disease, which is really very consistent with what it's observed in patients. So here again, we transferred the same identical mutation from one genetic background to another one, and we observed completely different phenotypes. This really means that there are other factors that are really regulating the disease severity, and we want to identify them. So for that, we're currently running a yeast study. And we have yeast in the lab that are from different genetic backgrounds. Their genomes have been already sequenced. And we've been inducing the disease with a drug, which is an inhibitor of the NPC1 transporter. And here in blue, you can see cholesterol accumulation triggered by the U drug. We've been following several phenotypes. One of the most common and easiest is the growth curves, which really represents the reproductive fitness of the yeast. And here you can see that some strains, that they are very resistant to the U drug or to the disease, while others, their growth curves, they change completely. And we've been using some parameters of these curves, actually the slope. as a trait. And here we have segregants. These are daughter cells which have been genotyped of the different combinations of the yeast strains. So we took the most resistant, we took the segregants of the most resistant versus the most susceptible strain, and here we are showing this parameter of the growth curve on all the segregants, and we have performed a linkage analysis, and we got two very significant spots. Currently, here there are around 20 genes in each spot, and we're currently narrowing it down with different techniques to really identify which is or are the genes that are involved in this differential response. I must say that we're very excited about using these kind of strategies, using the mice or the yeast here, because it also provides us combinations of genes. And usually when you're running conventional screenings, you're, for example, knocking out gene by gene. Well, here we get combinations of genes. And we've been also analyzing the cellular phenotypes. For example, here we've been staining the vacuole, which is the equivalent of the lysosome in the yeast. And you can see here that, for example, the strain 407 is completely resistant to the changes in their vacuoles, while this other, it's it changes completely. And the beauty is that there's no correlation between the intracellular phenotypes and the growth curves. So we're also mapping genes related to each of the different phenotypes. Okay, so now we're coming back to mice. I always like mice, and I was very interested in really looking for novel therapies for NPC in mice. So I treated really the mice. These are the NPC mice in the C57 background, which they live like for 4 weeks that I already showed you, with rapamycin. And with this drug, I managed to double the lifespan of the mice. However, when I treated the guys that live for longer times with the drug was toxic, and the animals died younger. So here again, let's talk. We're talking about the same mutation, the same drug, different genetic background, and we got completely opposite effects. For me, this is wow. And currently we're starting to use this yeast platform to really identify the genes that could be related to this differential response to the drug. So now the last couple of slides, I would say that in the last 10 years or so, it has been shown that lysosomal dysfunction plays a critical role in Parkinson's disease. And actually, gene variants in the same gene that triggers Gaucher disease is a major risk factor for developing Parkinson's. And also there's cell biology experiment, biochemical experiments linking the lysosome and PD pathogenesis. We summarized that in a very nice article. So like in every disease, not every patient, in Parkinson's, not every patient experiences the symptoms in the same order or at the same intensity. And the variable symptoms include motor disabilities, sleep abnormalities, and reduced lifespans. So I talked with with two collaborators from University of Chile, and we designed the following pipeline. My friends are using different panels of flies, Drosophila, which again, these are inbred fly lines. And we've been inducing a PD glycolysis with a drug, which is called rotenone, which induces Parkinsonism also in humans and in mice. And we've been focusing actually in two different phenotypes, motor problems and sleep abnormalities. So we've been analyzing several lines of flies treated with rotenone. We've been able to identify already some genes and gene networks related to the climbing and sleep problems. And currently, last week actually, we're very excited. We just got human data from patients with Parkinson's and sleep abnormalities. And this is something that we're doing in collaboration with Ziv Gan-Or from McGill University in Canada. And we will start as soon as possible. You know, we are in lockdowns right now in Santiago with the validation with humans. So the idea is actually then, of course, try to stratify patients according to variants in the genes or networks identified, and of course to develop drugs that now it will be symptom-specific drugs. So it will be for a patient with Parkinson's. If the patient also has, for example, REM sleep abnormalities, then we will give them the drug A. Again, we're very excited because we're managing really to merge the animals with the human studies in order to identify the best drug candidates. So just to summarize, I told you today a bit about the mouse GWAS that identify loci underlying the different clinical signs in mice that may serve as biomarkers. And also it helps us to design novel therapy for neuropathic Gaucher disease. I showed a mouse system genetics for uncovering regulatory networks of the lysosome. I show you a yeast in vitro precision medicine platform for uncovering modifier genes, which can be used also for pharmacogenomics. I show you that the response to a treatment, in this case rapamycin in NPC, was genetic background dependent. And at the end, I told you a bit about a fly GWAS that we're running, which is revealing modifier genes of PD symptoms in humans. And having said that, I must acknowledge I have a very good group of friends and collaborators from all over the world. My lab team, really, I mean, these are outstanding scientists, and I'm very, very excited about that. The funding agencies, well, the FLY Parkinson study, it's funded by the Pew Charitable Trust. We are part of an international consortium of the European Union called LysoMod, which aims to identify genetic and chemical modifiers of the lysosome, and also the funding from the Chilean government, which is equivalent to the R01s in the States. If you have any questions or whatever, here